specific examples to say, well, this project is really good because we can use it. Whereas, maybe what you're doing on this other device or parts doesn't interest us all that much, right? And obviously, if we talk about test beds, so a test bed that makes a chemical that can be used to make a biofuel would be much more important to a company that's into that. So, for example, Verenium, which is one of our members, than it would be to say a company that makes an antibiotic that isn't interested in biofuels. And so clearly there are differences as to where they think we should put our energy to benefit them. So we have to balance that as well. But clearly it's a great relationship because you get people sort of at the cutting edge of what's going on in industry, involved in our work, because they come to our retreats, they see our projects, they feedback, they interact directly with the postdocs and the grad students, and actually also at the next retreat we're planning are going to present what they do and what they think we should do in a more direct way. So clearly there is this working relationship, which is new to most people who practice science in academic laboratories. And I think that for a lot of students here who are going to end up in industry, it's their first look at how it functions, and it gives them a kind of advanced look at what they be seeing when they move into that sector after they're done.
So quickly, what did you study in throughout your education?
Well, you asked me what I studied throughout my education. It may not even be relevant because when I took my PhD, the tools and understanding of molecular biology hadn't been developed. I mean, when I first was looking at DNA, we were running gradients. There were no gels. There were no restriction enzymes. All of that came during and after and later. So if we wanted to move DNA around, we didn't take it out of cells and put it back. We had to move from one organism to another by doing standard genetics, which was conjugation. And I worked on bacteria, so if you wanted to do genetic mapping, you did it the hard way. You had to move DNA using a vector, but a natural vector. You didn't clone it. You just asked one cell to deliver it to another, or you put it on a virus or a phage of some kind and moved it that way. So it was much harder, much longer, and your progress was much more restricted. You couldn't do as much. But in essence, I studied the regulation of the breakdown of arabinose in E. coli, the metabolism, and how those genes that were involved in that breakdown were regulated, positive control system. From that, and not from my work directly, but just to show you where that went. So now, as a common vector for driving expression of genes, the arabinose promoter is used in a standard way. The regulators are used. So this P-bad promoter is a standard promoter used in molecular biology and synthetic biology. It came out from that work that happened, 30 years ago or longer. And so a lot of the elements of practice today derived from, obviously, the work that was done before that. And I worked on the arabinose system. As a postdoc, I worked on the replication of coli-1. And if you can take a look at that photo up there, and you can even show a picture of it if you can, because that's an electron micrograph of stages of replication of the plasmid coli-1, which forms the basis for the vector, the puck vectors, because it's the same replicon that's used in a standard way for molecular biology. So I was studying replication by doing electron microscopy and purifying DNA on sucrose gradients or cesium ethidium gradients. Never ran a gel in those days because we couldn't cut the DNA. Actually, we cut it once with EcoR1, the first enzyme we got, but we had to make it ourselves, actually. Actually, we had a collaborator make it and give it to us, Herb Boyer, who actually discovered that enzyme back in the 70s, and we used that to map the origin of replication. But my background was on regulation and on plasmid replication. And that actually served us well going forward because we developed vectors. As I started my career in industry, we tried to develop vectors, not for E. coli, but for organisms that were industrially important. For example, bugs, bacteria that made antibiotics. These are streptomycetes. So we, along with a number of other labs, notably the lab in England, Sir David Hopwood's lab, who developed a number of vectors for streptomycetes, along with a lot of other tools and basic understanding. We used that work and our knowledge and experience to try to do the same thing in organisms that my company was interested in. Organisms that made different antibiotics. And so each company had their own favorite organisms they worked with. They were related to each other, but different enough. So whereas Hopwood's lab worked on Streptomyces silicolor, which was like the E. coli of the Streptomyces world. It made antibiotics, but nothing that none of the antibiotics that we or other commercial companies were interested in. So that background I had as a grad student and postdoc served well to bring molecular biology into the drug industry to advance the production and the discovery of novel antibiotics. And that lasted for a long time.
So I know you went in June to this symposium on synthetic biology in Europe and the possibilities there. Could you say anything about the striking differences between America and Europe in relation to synthetic biology?
So you asked me about what's going on in Europe in synthetic biology and about a meeting I attended in Brussels, actually, in June, around synthetic biology in Europe. Well, there's an organization in Europe. It's not really an organization, it's an initiative. They call it TESI, towards a European strategy for synthetic biology. That they call that TESI. I'm not sure what the Y stands for, but anyway, it's TESI. And that comes out of the EU, which is funding this initiative, overseeing it. And there's a lot of money there to spend on this. And of course, in Europe, it's a lot of countries. The EU is composed of 10 or more, maybe 15 countries that sponsor, that give money to various initiatives, and that's administered by what they call the EC, the European Commission, much of which is headquartered in Brussels. And so that agency is funding this initiative towards developing this strategy. But the funding of projects in synthetic biology will come from a variety of agencies that are actually traditionally funding science anyway in Europe. So in England, they have funding agencies, France, other ones, and they're trying to sort of organize a strategy for funding so that you would have then funding projects somewhat like Synberg, where you would have a vision